this part through this cell bilipid layer. And that brings us to transports. How do things get across the cell membrane? Well, before we get into the specific types of transport, we need to talk about two broad categories: passive transport and active transport. Passive transport is transport that goes down or with the concentration gradient. In other words, if we're going down the hill, if we roll this ball, we're not going to have to spend energy to get that ball to go down the hill. It'd be a passive. It's going from areas of higher concentration to areas of lower concentration. Now in this case I've represented it as a hill, but we need to think about the concentration of molecules when we determine whether it's passive or active transport. Active transport, conversely, is going against the concentration gradient or up the concentration gradient. To get this ball to go up the hill, we'd have to spend energy, and cells spend energy in the form called ATP. Let's look at some specific types of passive transport, and the first is diffusion. Diffusion is the movement of materials from areas of high concentration to areas of low concentration. It does not cost energy. It's going down the hill. Down the hill. So in this picture, when we look at these yellow dots, whatever they may be, whatever molecule these represent, we see that outside of this cell there are a lot more of them than inside. So we have a higher concentration outside than in. So these yellow dots want to move which direction? Into the cell or out of the cell? That's right, they want to move in, down their concentration gradient. So if we were to represent this movement of these molecules in this direction, we'd call that diffusion. They'd be moving down their concentration gradient. And grab some of these guys. They'd diffuse across down their concentration gradient right through the lipid layer. And we call that diffusion. Now, question, what if these yellow dots represented water? If we let these yellow dots represent water, then this movement, this diffusion of water across the selectively permeable membrane has a special name. We call that osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. It's a very specific definition, and since it's diffusion, it's by definition passive. It's moving down the concentration gradient. Now, what about these down here, these positive charged ions? If these ions, if we look at it, which way do they want to move? Where is there a higher concentration, inside or outside? Well, it's very obvious to see that we have a lot more of these red positive charges inside, so they would like to move down their concentration gradient, which in this case would be out. But we know that ions have a difficult time getting through the bilipid layer. But if there's a channel provided for them by a protein, then we have a path for these ions to move down their concentration gradient. Now they're being helped by a protein, however, it's still passive, and we call it facilitated diffusion. It's the diffusion of materials with the help of channel proteins. It's still moving down the concentration gradient, so it's still passive. What about active transport? Can we move materials against the concentration gradient? For example, can we move these molecules out, even though out is going up the hill or against the concentration gradient? And the answer is we can, but it requires active transport. Let's see how this might work. We have some special gate proteins here that can physically change their shape, and these molecules can be moved across the membrane with the aid of these proteins. Now, that movement is going to require the input of energy. So let's pull these guys back in here and back it up and show the input of that energy. Let's follow what might happen. Molecules move in, we get an input of energy from ATP, which causes the proteins, again we call this active transport. It requires ATP and the help of transport proteins. Now what happens if the molecule or the particle we're trying to move in or out of the cell is this? Meaning on scale it's much, much bigger. It's obvious that this is never getting through the cell membrane. It's never going to make its way through even one of these channels. It's just not going to happen. So can we get large particles in and out of the cell? And the answer is yes. So I've changed the scale here, and here's our large molecule, And the question is, can we get something of this scale through this membrane? And the answer is through endo and exocytosis, yes. The movement of large particles in or out of the cell, getting wrapped up in a vesicle, into endocytosis, out of exocytosis. So let's look at some pictures. First, let's look at endocytosis. Let's say that some droplet falls on the surface of a cell and slowly indents further and further into the cell membrane. And it actually gets wrapped up in a piece of the cell membrane, which we call a vesicle. We call this form of endocytosis pinocytosis. Another form of endocytosis, called phagocytosis, the cell moves itself, changes its shape, and wraps around the cell particle, or the particle, maybe it's a piece of food, and eventually engulfs it into a vesicle. The opposite of endocytosis is exocytosis. It's basically pinocytosis in reverse. A vesicle moves to the side of the cell membrane, joins with it, fuses its vesicle with the cell membrane, and expels the particle out of the cell. We just have to remember scale. If we zoom in to this line, we have to understand that this black line represents this phospholipid bilayer that we've been zoomed in on. So that large particles can get in and out of a cell, but not through the cell membrane, yet wrapped up in a piece of the cell membrane called a vesicle. Now that wraps up our video on the properties and